when looking at the Krebs cycle is a cyclic process where one precursor molecule is being regenerated before it then goes and passes back around into the Krebs cycle. So we start off with the coenzyme, acetyl coenzyme A, which is bringing pyruvate that has been previously formed in glycolysis to the Krebs cycle. We then go and decarbonize it. So at this point, we go and we form a molecule of carbon dioxide. We also go, as we decarbonize it, we also go and form reduced NADP. So this 2-carbon molecule will then go and combine with a 4-carbon molecule to go and form citrate. Right, the 4-carbon molecule is oxaloacetate. Right, so we're forming a 6-carbon molecule, which is our citrate. Right, the citrate is further decarbonized at this point, so another carbon dioxide is released. During its dehydrogenation as well, we also go and reduce another molecule of NAD+. This 5-carbon molecule again is further decarbonized. So it's further decarbonized. So we go and we release another carbon dioxide. At the same time, we go and we reduce another NAD+. The 4-carbon molecule can then go and phosphorylate ADP into ATP. So we're producing ATP at this point. This 4-carbon molecule is then rearranged by an enzyme into a slightly different configuration, and we go and we make FADH during this process. We then go and reduce another NAD+ to finally go and regenerate that oxaloacetate. So we go and that re-enters back to the start of the Krebs cycle and the whole thing starts again. Right, key things is that all of these reduce NAD and the FAD can then go and move into the electron transport chain, which is where the majority of the ATP in respiration is going to be formed.